Okay, this is going to be a pretty quick assignment to do. You'll be able to do it as you watch the video, and it's going to be the drawing of two receptor signaling pathways that are examples that we're going to be using for our G protein coupled receptor pathway examples. And the G protein coupled receptors involved, there are hundreds of them in our body that play really essential roles every moment of our lives, and so in our normal health and also disease states. And then there are orthologs of these receptor signaling pathways in every vertebrate animal and lots of invertebrates. So even though we have somewhat of a biomedical focus on our course because of cancer biology, this is true for all kinds of animals. And so it goes like this. We want to start out because we're going to use these examples to help us understand a couple of concepts of all cell membrane receptor pathways. And so this would include all the receptor pathways we're talking about, all the receptor tyrosine kinase pathways, and all the other pathways we're going to talk about. And so here we're going to have a cell membrane receptor, and this is the cell, and people like to call this the target cell. And when the ligand binds through signal transduction, the target cell is going to make a cellular response. And our favorite one going forward is it's going to be that the cell goes through the cell cycle and we stimulate cell growth. But there are thousands and thousands of different types of cellular responses. They all fall into these categories. Sometimes the cellular response includes changing gene expression. So that's one type, is we could have new gene expression. And that's something we've already seen before, how ELK can activate MYC, for example, under the scale, okay? So there's going to be new gene expression, and there's a general term for that type of response, and it's called a genomic response. So a genomic response means new gene expression. Then we'll see it in the first example of this video, and it's very common. There's a second option, and that could be the cellular response could involve non-genomic responses. So that's going to be things happening in the cell, but it doesn't involve new gene expression. And the example we're going to use is epinephrine, which is synonymous with adrenaline. So a really quick response to a tiger walking in a room, and we get excited and run away like that, right? So that is a good example because it's a very fast response, and it does take several minutes for genes to be expressed, and we need to move now when the tiger is looking at us, right? So but there could be fast and slow non-genomic responses like that. They just don't involve new gene expression. And then really the most common cellular response is both genomic and non-genomic responses together. And that's going to be our second example. So we have lots of examples already where we're focused on things like FOXO, and FOXO is a transcription factor, and ALK, and so forth. And so we'll have ongoing lots of focus on that. But we should be aware that there are really important non-genomic responses, and often it's both, okay? And so we're going to use this, and we're going to see that the adrenergic receptors for adrenaline, we'll call it epinephrine, those are going to be a non-genomic response. And the dopamine receptor we're going to talk about is going to be both. Again, we have lots of examples of genomic responses. We've already had some, and we'll have more, okay? So just general concept terms like that to help us organize broadly our understanding of cell membrane receptors, okay? And so here's G protein coupled receptor pathways, and it comes in as our second section. So again, the book starts out with nuclear receptors. They're not cell membrane receptors, and it goes on to these other categories of cell membrane receptors. G protein coupled receptors is the first section. So our book does a good job introducing cell membrane receptors and signal transduction. So there's more kind of language in that section that's introductory and sort of mapping it out. And so we'll be able to put that together with these two direct examples. And so our first one is going to be the adrenergic receptors. They're the receptors for adrenaline. And you don't need to learn that name for this course if you don't want to. You could learn epinephrine, or if you're familiar with this old term that's synonymous with epinephrine is adrenaline. It's the adrenaline epinephrine receptor or adrenergic receptor. Just go with what you might already be familiar with that. And so the book introduces G protein coupled receptors. And here's just a schematic diagram of the primary amino acid sequence. And the book just wants to bring up that it was easy once scientists were able to start to sequence DNA and look at the coding regions of genes. It was easy for them to identify G protein coupled receptors because they pass through the membrane 7 times, and so that makes for them for having a distinct primary amino acid sequence because they have 7 patches of about 20 amino acids that are very hydrophobic. So it was easy, and so that's just kind of a historical note that the book really wants to share because all of these were discovered in the last 50 years during the author's adult career in science. Okay, and so then the book carefully introduced the idea of signal transduction. So it says, here's an example of signal transduction when we get the ligand binding to the G protein coupled receptor. In our case, this is going to be the adrenaline receptor, the epinephrine receptor, the adrenergic receptor, and I think I'll just call it the epinephrine receptor. And so what happens is the receptor gets activated. And in this case, the first step for G protein coupled receptors, because that's their name, is they activate a G protein. Now, this we're going to call, because it's a trimeric G protein. So that's different than the monomeric G protein that we are learning about called RAS. RAS is involved in a different kind of receptor signaling pathway, right? That's the receptor tyrosine kinase pathway. Okay? So this is the G protein coupled receptor, and there's the G protein that's coupled with the receptor. So all G protein coupled receptors start with this. And they have a trimeric G protein, and is 3 subunits, and they each play a role. The big emphasis is the alpha subunit, because this one is always bound to either GDP, or when it gets activated, it gets exchanged and is bound to GTP until that enzyme is hydrolyzed and broken down back to GDP and it's inactivated. So G protein, because it's always bound to either GDP or GTP, and when it's bound to GTP, it's active. When it's bound to GDP, it's inactive. Okay, so then what do activated G proteins do? And this is just one example, and it's the example of the epinephrine receptor. When the tiger walks into the room, our body releases the epinephrine hormone, and it binds to lots of cells, and the cell we're going to imagine is the muscles. So our muscles respond to epinephrine. Let's get out of here really fast, okay? So the G protein coupled receptor is activated, the G protein is activated, and in this case, it binds to an enzyme that catalyzes ATP to be converted to cyclic AMP. And cyclic AMP is a second messenger. And we've already met some second messengers. Second messengers are just kind of a loose term for some small molecules that are involved in signal transduction pathways. And this is a famous one. It's often involved in G protein coupled receptors pathways. It's prominently involved in the epinephrine receptor pathway. That's our example. So this will be part of our drawing. And this enzyme is called an adenylyl cyclase. It's also called adenylate cyclase, and it's also called adenyl cyclase. Those are all synonyms, but it's just an enzyme that catalyzes the synthesis of cyclic AMP. Here's the book in that second section, carefully introducing the concept of the G protein. Active when it's bound to GTP, and inactive when it's bound to GDP. We're already familiar with that because we know RAS and how RAS works. But just remember, these are trimeric G proteins here. It's a really different category, but activated in really the same kind of style of way. So then the book also wants to show you the concept of this is ATP and this is cyclic ATP, cyclic AMP rather, and that's the second messenger. And we don't need to know the structures of these. Not in your wildest dream do we need to know this, the structure of these. We just need to know that adenylyl cyclase is activated by a G protein and stimulates the synthesis of this cyclic AMP second messenger that can float throughout the cell, throughout the cytoplasm, again, in our case, of the muscle. Okay? And so here's the book showing the full response, and this would be in a muscle, and this time they are naming it epinephrine binding to, call it the epinephrine receptor, activating a G protein, binding to adenylyl cyclase, stimulate the synthesis of cyclic AMP. And cyclic AMP can do lots of things. One thing it does is it binds to a protein called protein kinase A, which is often abbreviated PKA. And it's called protein kinase A because it was kind of the first, I think, protein kinase ever discovered. And remember, there are 500 kinases in our body. So, you know, this was discovered back in the 1950s and 60s like that. And it looks like this. It has some regulatory subunits that are inhibiting the activity of the kinase. And when cyclic AMP binds to the regulatory region, those subunits are release the catalytic domain or catalytic subunit. Catalytic, should call it catalytic subunit, not domain. It's a full protein. Okay, and that kinase is going to use ATP to catalyze the phosphorylation of target molecules. And in this case, it's going to activate another kinase, and the target is an enzyme. And we don't need to know the names of these, and we don't need to know the details of their activation. What we want to know is just that epinephrine binds to the epinephrine receptor on muscles, activating a G protein, activating adenylyl cyclase, synthesizing cyclic AMP that activates PKA, and that leads to the breakdown of glycogen. So glycogen is a polymer of glucose, and it is used by muscle cells and lots of cells in our body to store sugar. And when the tiger walks into the room, our body releases the hormone epinephrine, and that helps us run away because we get a sudden burst of sugar in our muscles and our brain and other tissues, but in our muscles in particular, that's going to give us a burst of energy to run away. So this is going to be broken down right away and produce ATP for muscle contraction. And it just lasts, for a minute like that. And it's like when you eat a big slice of cake and you get a sugar rush, right? Like that. Well, this situation is we've got that sugar already stored in our body, in the cells that need it when we got to get away from a tiger. And so this is the drawing for the first drawing in our assignment. So do this drawing and label this. Again, we want to label epinephrine receptor, G protein, adenylyl cyclase, cyclic AMP, PKA, and glycogen making sugar. These, intervening steps, not such a big deal for us. If you can include those on our drawing and be ready to include that drawing on an exam, that'll be great. And this is a great example of a very fast non-genomic response. And it's kind of the most memorable one. You could remember this for the rest of your life. This does not involve new gene expression. There's no transcription factors and target genes involved in this response. It happens really fast. In just a minute, you're just like, I'm out of here. Well, less than a minute, right? It happens really fast. Okay? So that's our first example and our first of two drawings. Our second example is the dopamine receptor, and this is going to include both, we'll talk about first, a genomic response and a non-genomic response. Okay, so we're going to see some receptors, and the dopamine receptor is a gene family, so they're given special numbers and letter designation, and we're just going to remember it as the dopamine receptor. And here's the first part of your drawing. We have to make a hybrid drawing where we add something to it, because it's going to be both this, a genomic response, plus it's going to include a non-genomic response. So dopamine is a very famous neurotransmitter, and so here they're labeled as a hormone because dopamine is a neurotransmitter inside our central nervous system, inside our brain, and outside of it, it's not really a hormone, it's a paracrine factor, it's a local signaling factor. So its biology is complicated, but the biology that most of us are already familiar with is dopamine is in the reward center in our brain, and it functions in neurotransmission when we're feeling happy and enthusiastic and receiving rewards. This receptor pathway is activated in nerve cells, and nerve cells are called neurons, and so we're looking at a neuron in our brain. And again, all animals have this, right? All animals have this reward response in their brain, okay? And it turns out it concludes both a genomic response and a non-genomic. And our book shows a really nice example of what is the genomic response. Now, other G protein coupled receptors use this pathway. So our book is not being really specific on this being dopamine, but this is a great example. Our example is this is the dopamine receptor, and dopamine in the brain will bind to the dopamine receptor, and it will activate a trimeric G protein. And this trimeric G protein happens to again activate adenylyl cyclase, and we synthesize cyclic AMP. Now, these G proteins in various G protein coupled receptor pathways, they can activate a lot of different stuff, but both our examples and drawings happen to include adenylyl cyclase. Okay, great. In this case, the cyclic AMP again binds to the regulatory subunits of protein kinase A, or pKa, and it releases a catalytic subunit. Okay. Now, before we saw in the other example of epinephrine that phosphorylated ultimately enzyme to break down glycogen. In this example, the pKa catalytic subunit goes into the nucleus and phosphorylates a transcription factor called CRAB. And CRAB activates gene expression. So in our brain, dopamine can activate gene expression. It can also act, we'll see, as a neurotransmitter. So it's going to make cyclic AMP and be a neurotransmitter. So it makes both a genomic response and a non-genomic response. And CRAB transcription factor is just an example that we can add, and these would be target genes that are activated by CRAB. People usually refer to these as cyclic AMP target genes. And they all have an enhancer that binds CRAB. And people like to call this enhancer a cyclic AMP response element. And I think it's on the next slide that I have the name, and you can just remember CRAB, but there's the full name of CRAB. It's the cyclic AMP response element binding protein. Because scientists in history first identified cyclic AMP target genes, then they used those methods that we talked about, like deletion studies and nuclease protection studies, and gel mobility shift studies and so forth, purification, to first identify the enhancer of cyclic AMP responsive genes in this genomic response. And then once they've identified the enhancer, let's find out what transcription factor binds to it. And they call that transcription factor CREB, cyclic AMP response element binding protein. Now, you can just remember CREB, but it's satisfying to learn the nature of its name, because its name makes sense based on how we know people discovered transcription factors in history. Okay? It just fits into our list of transcription factors, being it gets, in this case, activated by phosphorylation. Phosphorylation by pKa in this case. So we added two examples of FOXO that gets inactivated by AKT, and we added two ELK that gets activated by ERK, and we get CREB that's activated by pKa. So all these are regulated transcription factors switched on and off by kinases. Great. All right, so that's part of your drawing. And the second part of your drawing is the non-genomic response. So the dopamine is a neurotransmitter. It causes action potentials. It causes immediate responses like thinking and emotion events in our central nervous system. And that's because this cyclic AMP, if we just drew the cell membrane further, this cyclic AMP also binds to proteins on the cell surface that are ion channels. And the way our neurons communicate between each other in our brain is by changing the concentration of ions like sodium and potassium and calcium across the cell membrane, and that leads to action potentials, or essentially the electrical communication pathways through our brain. So that's going to be our non-genomic response, and our book doesn't show that in one figure, but this is your drawing. It's just to show, in addition, the cyclic MP going over here and binding a ion channel protein, allowing for neurotransmission. So two responses, okay? So let's just look at example of the genomic response medically. So here again, you know, the D2 receptor, we know it as the dopamine receptor. Again, it's got names and numbers because of the gene family. Activation of CREB through phosphorylation of the transcription factor and binding to cyclic AMP target genes provides a potential mechanism for effective antipsychotic drugs. So just adding this as an example of the relevance of the dopamine receptor in health and disease. And I mentioned before that well over half of all drugs are understood to affect G protein coupler receptor pathways. So we talk about just medically significant class of receptors, and that includes the dopamine receptors. They can be affected by drugs that can be antagonists or agonists of the receptor itself, or can affect that pathway. Okay, so you don't need to know this in detail. It's just a sidelight, right? The assignment was to draw and understand the two pathways. That's the genomic response. Here's another example of the genomic response. A dopamine-induced gene expression signature relates neuronal function and cocaine response. Okay, and so this is a big area of research. This would be genomic responses that are involved in drug addiction and other kinds of addictive behavior. And of course, it's a much more complicated story than this little slice of introduction, but drugs of abuse elevate dopamine levels in a region of the brain and alter transcription programs to promote long-lasting synaptic and behavioral adaptations, like addiction. And there's a good chance that you've heard some about the biological neural basis of addiction, and this is part of it. There are more pieces to it, but part of it is that the genomic response is affected. we have long-term changes in gene expression in our brain in response to addictive drugs and behaviors. Okay? So just examples of that. And this is just a figure that shows both of these pathways, both the genomic pathway and the non-genomic. The book doesn't show these in one figure. So there's our dopamine receptor. Again, funny name, you don't need to know. Just, hey, there's the dopamine receptor. So it's going to be binding the ligand dopamine. And when that gets activated, here's our trimeric G protein. And there's the alpha subunit that when it gets exchanged to bind GTP, it's going to activate adenylyl cyclase, like the figure in her book. That's going to convert ATP to cyclic AMP, and the cyclic AMP can activate protein kinase A, and that will lead to the phosphorylation of CREB, transcription factor, and new gene expression. That's our genomic response. So that's showing, again, all of that is showing in this figure. And this is just going to add to your drawing the last piece, which is the non-genomic response. And the non-genomic response in this case is a good and simple non-genomic response. Cyclic AMP can bind to pKa, activate that pathway you already have. In addition, it can activate this non-genomic response, not new gene expression, another kind of response. The cyclic AMP binds to this ion channel on the cell membrane, and that's going to cause a change in the concentration of calcium, potassium, sodium across the membrane through different ion channels, and that's going to lead to action potentials, and those are the signals that go through our brain and our nervous system. And so that's the second drawing, like that. Okay, so we're all set with the assignment. I just want to show you that dopamine is involved in non-genomic responses in many functions in parts of our brain. For example, learning, reward, motor control, emotion, and cerebral functions like planning and so forth, like that. And so that's again a role in neurotransmission, direct, rapid, really fast non-genomic responses. And so in the neuron, the brain cell, is a great example of both genomic and non-genomic responses. So again, all cell membrane receptors can either lead to genomic responses, and we have examples of that, like MYC and ELK and so forth, and we'll have more. And then non-genomic responses, and this might be our only example in our course, but there are many, 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 many like that, or both. So MYC, great example, genomic response. Epinephrine receptor, great example of non-genomic response. And then dopamine receptor, and the G protein coupled receptor pathway that dopamine activates. Both activation of CREB and new gene expression, and activation of ion channels. Great. And so just to show you, the book also talks about the human sense of smell. That's a detail. And maybe it would be 1-2 point question on exam, but that wasn't the example we wanted to really focus on. It's all our smell receptors. So when you smell cinnamon, the actual molecule binds to a G protein coupled receptor in the lining of our nose, activates a G protein, activates, in this case again, adenylyl cyclase, and then activates a ion channel. So a really similar story to that non-genomic response in the dopamine receptor pathway, right? So we don't need to use that as an example, but just to be aware, our book talks about it, and it's a kind of a funny and interesting one, right? It's a really fast one. We smell it right now, right? So it's another neurotransmission one, opening ion channels in the cell membrane, like that. And then our book no longer has this, but in earlier editions, I just want to show you that that's also how our eyesight works. So in this case, the G protein coupled receptor is holding on to a light-sensitive pigment called rhodopsin. And the light just changes the structure of the rhodopsin, and that activates this receptor. And look what goes on here is that we get an activated G protein, of course, because it's G protein coupled receptor. And then here's a different kind of enzyme. And again, not in our book, you don't need to know this, but another non-genomic response, right? Your vision, does that happen fast? Oh yeah, that's happening really, really fast. So that's just, it's startling to appreciate how fast these receptor pathways can occur when they're non-genomic. Not all non-genomic responses are fast, but that's a great way to introduce them because genomic responses take a few minutes. So of course our eyesight and our sense of smell are non-genomic G protein coupled receptor pathways responses because they're really fast. Okay, so we're all set with that. 2 drawings, and in the second drawing, remember that you have to build that hybrid image like that of the dopamine receptor. That's first a genomic response is well represented, and we just want to emphasize to get that second one in, and the second one is a very simple addition to your figure. Like that is just to have some of the cyclic AMP come over here and activate that ion channel. So 2 pictures, and pretty brief, but you just want to be solid on this material because it's such a famous and significant class of receptors, the G protein coupled receptors. Great, thanks.